Biological network representation and analysis has become a tool for many biologists and bioinformaticians, as these interactions graph allow us to map and characterize different pathways and predict the function of a protein. However, given the size of and complexity of interactome dataset, extracting meaningful information from interaction network can be a daunting task. Many tools and approaches can be used to build and represent and analyze biological network. A biological network is simply a collection of connected objects. Here, objects refer as nodes and vertices, and usually drawn them as a point. Connection between the nodes are referred as edges and usually drawn as line between the points. In mathematics, a network are often referred as graph. In this picture, A, B, C, D with circular are nodes and the connected lines are known as edges. Some basic features, the structure properties of biological network, the size. The size of a network depends upon the number of diameter. It is the greatest distance between any pair of nodes. Density of a network describe about the proportion of the connection. Average, or we can say that a characteristic path length defines as the average distance between all the pairs of the nodes. Shortest path, a path between two nodes such that the sum of the distance of its constituent edges is minimized. Degree, it simply measures the node of the centrality, or we can say that how well a node were connected in the network. Degree distribution, you can calculate by counting how many nodes have each degree, and we can form a degree distribution. Centrality, centrality gives an estimate how important a node or edge is for the connectivity or the information flow of the network. Degree centrality, number of edges adjacent to a node or number of edges connected to a node. Closeness centrality is the useful measure that estimates how fast the flow of information would be through a given node to other nodes. Betweenness centrality, nodes with a high betweenness centrality are interesting because they lies on the communication path and control information flow. Its value varies from 0 to 1. Clustering coefficient is a measure of a degree to which nodes in a graph tends to cluster together or how well nodes were connected. Its value lies from 0 to 1. Network motif, patterns that occur in a real network significantly more often than in a randomized network. Hubs, nodes in the network with the maximum number of connections. Hub serve as the common connection mediating the shortest path length between other genes. Modularity measures the strength of a division of a network into modules, group, or clusters. Clique is a subset of a node in which every node is connected with every other member of the clique. Modules, modules are the exchangeable functional unit in which the nodes, it can be a protein G, do not have interacting in the same time or space. In this picture, you can see that the graph with the purple and blue nodes, where the blue node represent the highest degree as it connected with maximum number of nodes in the graph. And the centrality analysis can be studied with the help of degree. The motif shows represent the pattern that occur in a real network significantly more often in the randomized network. Click means how well you are connected within the network, or where others are the shortest part, and the search motive. The last is graph with the gray color represent the network community. Types of biological network. Different types of data will be used to produce different general networks, characterized in terms of connectivity, complexity, structures, where the edges and the nodes are potentially convey the multiple layers of information. Net biological systems are often represented as a network, which are a complex set of binary interactions or a relation between the entities. Where the network biology in which we it allow us to represent and analyze the biological system derived tools through graph theory. Some of the important common type of biological networks are protein-protein interaction networks, metabolic networks, genetic interaction networks, gene transcriptional networks, regulatory networks, cell signaling networks. In this figure, you can see that A, B, C, D, E, F are the nodes with circular. The square box represent the metabolites. The orange color edge with an arrow represent the metabolism process taken place between A and M2. Between A and B, edge without the arrow represent the protein-protein interaction. Between C and B node, you can see it in associated shows that it are protein-protein connection also visible, and the green dash edge with an arrow represent the gene regulation. In node D and E, you can see the green color edge dash edge with an arrow representing a gene regulation and so on. Gene interaction network. Gene interaction network is a set of a gene connected by an edge representing the functional relationship among the genes. It occurs when the combined phenotypic effect of a mutation to a gene is significantly different from that of that expected. If if the effect of each individual mutations were independent of the other. In gene interaction, the nodes will represent the gene and the edges represent the unidirect. In through gene interaction, we see the interaction. It can be a physical expression, it can be genetic, etc. The tools and database that can be used for studying the gene interaction network, GEO, gene expression first David network analysis, gene string, cytoscape, etc. How to build a biological network? First of all, we have to search and retrieve the knowledge base from the database. It could be a David, it could be a GEO, it could be through NCBI, it could be anyone from the database. Then we predict the data from the genome sequence, where we study about the gene cluster, gene fusion, physical or co-evolution. Then we predict from the omics data, where we study about the co-expression, its classification, and we do the literature mining. Predict from literature, that is text mining, and then meta network from genome scale data analysis. GENEMANIA. This is the tool I have chosen to show you how the gene interaction network can be constructed and how it can be analyzed. As we can see the interaction networks by describing how they are generated, where they are stored, and how they are shared. We focus on publicly available interactomes network and describe simple way of utilizing these. The another tool I have chosen is the CYTOSCAPE. Is an open source and easy to use network visualization and analysis tool to first gather and visualize network. We have analyzed this network topology features and have to look at functional enrichment of the network nodes by the genes ontology database. The method described are applicable to a large scale network. This is the homepage of GENEMANIA. First of all, you have to collect the genes, the number of genes you wish to work, paste it on the gene box. Then you have to select your organism you wish to work, then click on the search box. Here I have selected some of the genes from different cancers, and In the arrow box, you can choose the attribute also, where you want to see the attributes, where attributes hash represents the pathways you or the other networks. You can choose the co-expression, localization, anything. Then this is the graph has shown in this picture, where you can change your layout according to you. The three dotted line, when you click on that, a docking window is opened where you can extract your data and the image from them. Multiple interactions with different colors will represent. You can select, deselect according to your wish. This is the same graph but with different layout. A zoom out picture of that graph. The gene represent with a circular node with edges on them. The second column represent the associated genes with black color, and the third represent the associated net pathways with the square gray box. The edges represents with the different colors, and the width of the edges also significant as the breadth of the edges represent the how strong the connection between the node and the associated genes. If we want to study a particular functions within the graph, then you can open up that function box with this window, and then you select your desired function. Then it will show you or indicate it within the graph with different colors and highlight them. And you can say that I have selected the regulation of cysteine type, and then it will show highlighted our DAGs, our genes, the nodes, and the associated nodes and the pathways. If we want to study a particular node within the graph, then you have to just click on that particular node, and it will show you that which are the nodes that are connected or associated with them, and what are the pathways that are associated with that. This is an example from that with any attribute you can download. These are the notepad data where the GENEMANIA shows the networks, where you can see the co-expression as a group, name, width, references, PubMed, source, and nodes. From this data, you can analyze about the network and which association, which nodes is connected to which pathways. Another tool, this is CYTOSCAPE. CYTOSCAPE is a tool through which we can visualize our network and analyze our network. In this, you have to first download your cytoscape and then install the packages you wish to work. For example, I have installed the cytoscape with a GeneMania package. You can use with strings or network analyst or anything. Then you have to select your database, your source, your plugin you wish to work. Then you have to choose your model organism, then paste your desired gene and then search. In the result panel, you can see the interactions, different interactions that are showing in the network. This is the example of that network. Genes with black color, circular nodes, associate genes with gray color, and the associate interaction. The red color represent the physical interaction. The purple color represent the co-expressions between the nodes and the associated nodes. So from the result panel, you can study your network, your gene, and the function. Here, when you click on the gene, you have your genes with different colors and rest of the associated genes with a black color. Then, if you click on the function, you will have the file with Q coverage value and GO annotation. Here, GO means gene ontology, where we study about a particular gene on the basis of biological function, molecular function, and molecular process, and the cellular component. True result panel represent about the clustering coefficient is 0.528, connected component is 1, network diameters, radius, centralization, shortest path. By all these parameters, you can study for your graph, or we can use these parameters for extracting the modules. For example, if we have a large number of nodes and we form a complex network, with the help of these parameters, you can extract the modules from them by choosing the by if you select the degree centrality or between a centrality. If you set your criteria that you want to have a nodes with a degree 1, then it will extract it according to them. Here is a example like between a centrality. A graph dot scatter plot is shown in figure where y-axis shows the between a centrality and the x-axis shows the number of neighbors. Here, between a centrality captures which nodes are important in the flow of a network, and it make a use of shortest path in the network. Molecular interactions are important to molecular biologists because to understand a gene or a protein's function and behavior, to predict the biological process of that protein with unknown function, they can help to characterize protein complex and the pathway. Interaction networks can be used as draft map to add details of biological process and pathway. These are the references